we'd like to take a moment to thank our sponsors.
Global climate change is already affecting the environment as we know it. The burning of fossil fuels has increased the amount of carbon dioxide in the atmosphere. Secondary pollution like smog and ozone are increasing. Droughts will be more frequent and severe. More intense precipitation events have resulted in extreme flooding around the world. From 2014 to 2016, the planet experienced the hottest years on record, which is quite astounding. Even more remarkable, the planet has continued this trend for the last 3 years as well. The impenetrable Doomsday Vault in Svalbard flooded as the permafrost melted around it. We experienced an intense hurricane season in 2017, and the National Oceanic and Atmospheric Agency has stated that the conditions are right for 2020 to be another difficult season. Carbon dioxide in the atmosphere and the planet's temperature are intrinsically linked, and the current carbon dioxide levels we have today are almost double what they have been for the last 800,000 years. The changes in the climate that we are currently experiencing are truly global issues. The regulations enacted by the United States had reduced air pollution. However, recent research has shown that the chemical precursors leading to increased ozone concentrations in the western U.S. likely have their origins in Asia. Additionally, the steps taken to reduce the amount of air pollution is reliant on continued regulation, which can change with administrations, special interest groups, and political will.
So how will a changing climate affect our ability to produce crops here in Illinois? Which of these challenges associated with climate change might a producer expect to face? How can we be better prepared to deal with these changes? We know that agriculture has always dealt with the variability in temperature, precipitation, pests, and disease. However, it is the rapid change in carbon dioxide and ozone concentrations in the atmosphere that we are less prepared for. This obviously creates challenges, but it may also be an opportunity to understand how plants respond to a changing climate while making agricultural production more resilient into the future. A team of researchers, technicians, and students have been trying to gain a better understanding of how these stressors will affect the crops we grow. Tucked into a small research farm located at the University of Illinois is the Soybean Free Air Concentration Enrichment Facility, better known as SoyFACE. SoyFACE is an open-air field laboratory for investigating the effects of elevated CO2 and ozone concentrations, higher temperatures, and altered soil water availability on crops. For 20 years, experiments at SoyFACE have examined the productivity, physiology, and genetics of crop responses to climate change in the field under replicated, fully open-air conditions, meaning the crops are grown in the field as they normally would be while only changing the microclimate. What makes this facility so unique? Let's zoom in and have a closer look at the technology. Early in the spring, just after the crops are planted, the researchers lead an ambitious effort to construct the FACE infrastructure around the 80-acre farm. T-posts are pounded in octagonal rings, and fumigation piping is hung. Small wind vanes and a series of sensors are installed to monitor the levels of CO2 and ozone within the rings. We can zoom into a single ring to see this technology in action. Each ring at SoyFACE is approximately 20 meters in diameter. The anemometer located in the center of the ring determines the direction of the prevailing wind. This information is sent back to a small control box, which controls a manifold, which ultimately releases either CO2 or ozone from the upwind side of the ring, allowing the wind to carry the elevated concentration of gas across the ring. The amount of the treatment is monitored in real time and varied to maintain a given set point. As the wind direction changes, so does the point source for the fumigation, allowing for a steady and continuous treatment throughout the day. The use of this technology at SoyFACE has allowed researchers to study the impacts of CO2 responsiveness and ozone tolerance in soybean and corn, and have revealed significant genetic variation in both crops. This suggests that targeted breeding could lessen the impacts of climate change. Additionally, transgenic strategies for improving crop performance in the future have also been tested. Why are we researching elevated CO2 concentrations? We care about rising CO2 levels for a number of reasons. The first is that it's the primary substrate for Rubisco. As CO2 concentrations increase, it outcompetes oxygen fixation and allows C3 plants like soybean to begin to close their stomata. Elevated CO2 generally improves production of important C3 crops like soybean, rice, and wheat by improving photosynthesis, biomass, and ultimately yield. Current CO2 concentrations are limiting C3 photosynthesis, as you can see in this CO2 response curve. The solid gray line shows the supply function of atmospheric CO2 concentrations. Where it crosses with the solid black line represents the operating CO2 concentration. For a C3 species like soybean, there are still photosynthetic gains to be made as CO2 concentrations increase. We care about ozone concentrations because it is a highly reactive oxidizer. As you can see in this cross-section of a leaf, ozone enters through the stomata, occupying the intercellular airspace, where it reacts and forms other reactive oxygen species. These reactive oxygen species cause cellular damage and decreases photosynthesis. Damage at the cellular level feed forward to reduce leaf area, biomass, and accelerate senescence, which in crops decreases yield. The photos on the right show some common signs of chronic ozone damage with programmed cell death, leaving a mottled appearance in tobacco leaves, and chlorosis or bleaching in soybean. Research conducted at SoyFACE has also focused on the interactions of an augmented atmosphere with altered water availability and elevated temperatures. As shown in this clip, we have developed a portable awning system which can be automatically deployed during rain events to cause water limitation stress in an altered carbon dioxide or ozone environment. Once the rain stops, the awnings are retracted, allowing the plants to continue their growth undisturbed, less the augmented atmosphere. A newer addition to the SoyFACE arsenal is the installation of the rainout shelter. This greenhouse comes with a retractable roof and side panels that allow the plants to grow to complete maturity in a fairly normal agronomic setting, but provides the researchers with precise control over every drop of water the plants come into contact with. SoyFACE also has means to increase canopy temperatures with portable infrared heating arrays. Although not shown here, these heating arrays have been deployed over soybean and maize in elevated CO2 experiments in years past, to understand how a changing atmosphere may interact with increases in temperature that is predicted in the future. For a deeper understanding of how these resources have been used, the research conducted at SoyFACE can be broken down into three major phases. Impacts. Simply stated, how do these crops respond to elevated CO2 and ozone environments? Interactions and gradients. How these crops respond when grown in these different atmospheric environments with an additional stressor, like drought and elevated temperature. And finally, adaptation. How we can move forward with the knowledge gained from these experiments to improve crops to a changing world. The first phase concentrated on the impacts of rising CO2 and ozone on soybeans and corn. The map on the left shows the experimental design during the early years of the project. The dots represent the ring locations and the colors show which treatment was applied and where. The green dots being the control rings, blue being the elevated CO2 rings, the red dots are the elevated ozone rings, and the yellow are the elevated CO2 and ozone rings. The second phase looked at how elevated concentrations of CO2 interacted with simulated drought conditions and elevated temperature in situ. The key innovations were developed by Drs. Andrew Leakey, Carl Bernacki, and Donort. Dr. Leakey's group developed an automated drought awning, which we showed before, to capture nighttime rain events during the growing season. The heating arrays that were shown were developed by Drs. Bernacki and Ort to maintain a 3.5 degrees Celsius increase in canopy temperature relative to an ambient canopy within both the soybean and maize CO2 rings. These same heating arrays were also used to investigate the effects of heat waves on soybean in both ambient and elevated CO2. An ozone dose response experiment was also performed during Phase 2 to obtain a better understanding of how ozone differentially affects soybean at varying concentrations. This study also looked at different soybean varieties and showed intraspecific variation. The photo on the bottom left of the slide shows an example layout of one of the elevated ozone rings. The third phase centers around adaptation. A lot of this current research builds on the knowledge gained in the previous years of the experiment and is in the process of being analyzed and published. We are utilizing advances in transgenic technology to make plants that will be better suited to fully utilize predicted increases in global CO2 concentrations by not downregulating photosynthesis or being better adapted to elevated temperatures. We are also using advances in genetics and genomics to discover genes and QTL regulating crop responses to elevated ozone. Here's a list of some of the publications that have come out of SoyFACE to date. Obviously, there isn't enough time to discuss all of these. For the remainder of this video, we will concentrate on a few of the findings that have helped drive the Phase 3 research at SoyFACE. Kristen Bishop, a former graduate student of the Ainsworth Lab, showed that there is genetic variation to soybean grown in an elevated CO2 environment. This graph shows the relative increase in yield of 18 cultivars grown during Phase 1 at SoyFACE. We built on this research in Phase 2 by taking a closer look at a high and low responding variety, LOTA, and HS93-4118, to see if we could parse out what was causing these different responses. Alvaro, a visiting researcher from Spain, concentrated on determining the mechanisms that drove the differences Kristen saw in her research. This figure shows how yield is correlated with three different efficiencies that are determinants of yield: light interception, radiation use efficiency or photosynthetic efficiency, and harvest index, the amount of above ground biomass allocated to the seed. We've highlighted the middle panel to point out that photosynthesis is a driving force in the yield gains seen when soybeans are grown in elevated CO2. Photosynthetic efficiency also appears to be a driver in the decreases we saw in the ozone dose response in Phase 2. This work was published by Amy, another SoyFACE alum. This graph shows the percent change on the y-axis and AOT40 on the x. AOT40 is a metric commonly used to describe the effects of ozone on vegetation. The control for this dose response would be to the far left, and the higher concentrations of ozone are on the far right. As you can see, Amy's research shows a linear reduction in soybean yields above background levels of ozone. This reduction in yield is driven by decreases in light interception efficiency, harvest index, and more dramatically, by photosynthetic efficiency, the light blue line. More recently, Tim Wharton, a former member of the Leakey Lab, and Nicole Chiquette, a former master's student in the Ainsworth Lab, have seen genotypic variation in photosynthesis in a diverse group of maize hybrids grown at elevated ozone. This graph has net photosynthesis on the y-axis and the 45 different hybrids sorted from lowest to highest photosynthesis in the control treatment on the x. This is shown with the white circles. The red circles show the photosynthesis of the same hybrids, but grown at 100 parts per billion ozone, a little more than double the concentration on an average day in the summer in Champaign-Urbana. The ozone treatment resulted in a 15% average reduction in net photosynthesis. We believe this research shows that photosynthesis is a key trait that describes plant responses to atmospheric change, and it is a worthwhile target for the adaptation of crops as we move forward. FACE has been an important innovation for studying how crops respond to atmospheric change, and will continue to be important as the planet continues to change. At SoyFACE, we are able to grow crops in the field under normal agronomic conditions while simultaneously simulating an environment in the future. The size and scale of this unique research facility has allowed the researchers to combine genetic questions with climate change experiments. New technologies and techniques will allow this research to take advantage of high-throughput phenotyping to gain a more thorough understanding of plant responses to climate change in hopes of adapting agriculture for the future. If you would like to find out more information about the research at SoyFACE, you can visit the SoyFACE website at www.soyface.illinois.edu. From there you can read more about the technology, see more results from the facility, and see a list of publications that are associated with the research facility. Thanks for watching.